folks, it's Chris here. Today we're going to be doing a mock DNA extraction. I'm here with Schiebelhut.
Hi.
Camera person. And basically I'm going to be doing a light explanation of what exactly we do when we try to extract DNA from animal cells, hair, skin, stuff like that. Because we've had issues lately with contamination in our lab, we're going to be testing the test tubes. So we're running a dry sample, meaning that we're just using water and our reagents, and we're not actually putting in hair or anything else like that. So we're going to see if we get DNA, which we should not at all. The two reagents we'll be using in this step are ATL, tissue lysis, and proteinase K. The tissue lysis basically tears apart whatever cells you're looking at, and it kind of exposes the DNA. But in that process, these little DNases, these tiny little proteins that will chew up DNA, will go rampant if they're not controlled by proteinase K. Proteinase K will chew up the things that chew up DNA. Because we're trying to look at DNA, we want it to be intact. So, previously I filled up these pre-measured vials, and I'll be extracting specific amounts into each tube, and hopefully at the end of this whole process we won't have any DNA in any of our samples. So we'll begin. We're starting off with 180 microliters of our buffer ATL. We have our ATL pre-measured. Get it close up if you want. Our test sample number 1 is marked. Being extra precautious because we do have contamination previously in the lab. I'm changing my pipette tips every single time I go through this, just to be safe.
So all the tubes and tips are pre-sterilized or autoclaved?
They are pre-sterilized and autoclaved, yes. And by doing that, we make sure that whatever little tiny living thing that's on here gets destroyed so that it doesn't interfere with anything that we're doing. Our pipette tips are also filtered. You can see there's a little silicate gel here, and that prevents any DNA from getting in places we don't want it to.
So what kind of DNA are you interested in extracting?
Personally, I'm interested in Paragnathus DNA. Those of you who are local to the Merced area know the vernal pools reserve we have just outside of campus. And out there's a species that only lives in this particular region of the state. And nobody has really looked into the genetics of Paragnathus. So my job in this experiment is to find out exactly how different they are from other species living out across the country. So hopefully, if we can figure out our contamination issue that's been plaguing us for the past couple months, we can find out how similar these species are to other ones throughout the state.
That sounds cool. And how do you use DNA to tell you that?
We can do all kinds of fun stuff, like making trees, looking at their actual DNA and seeing how similar it is, and it's all through computer programs and models. You want to go into some detail, Lauren?
That sounds pretty good to me.
Sounds pretty good. Alright, so now that we've put our 180 microliters of ATL, we'll chew up our DNA. But before it gets too far, we want to add our proteinase K, just so it doesn't run rampant and destroy everything we're trying to work with. Changing our pipette tip sizes also, because they do have a max limit, and if you do go up to the well, you will ruin your sample. So when pipetting, you also want to make sure that you're going really slow and being extra precautious not to splash or get too much. sometimes the pipette tips don't work, so you just throw them out and get another one.
So what's the difference again between the green and the clear tubes?
The green tubes, we suspect, have DNA contamination on them, because the green tubes were originally in these large bags, and people tend to just grab one tube at a time. I'm guilty of this as well. So, exactly. So what we should do is pour them onto a clean surface area, one at a time, and pull them out. So this bag is brand new. I just opened it right now. So we know it's never been touched. We know that it's sterile. So all these blue containers here, these 4, should have no DNA on it, whereas these might have some.
Do you know what kind of DNA is contaminating?
Currently, I do not. I'm assuming it might be from other samples. If, for example, you draw out some Paragnathus DNA and you make a mistake, and it rubs your hand against the pipette, and you go to drop it, and you reach into the bag with DNA from one individual, you pull out a tube, touch the tube, and then that individual's DNA gets into a tube where it's not supposed to. So just trace amounts of anything anywhere on your body can really cause issues with contamination. So when working with DNA, we really want to be very, very, very careful to not mix up our samples.
So then the plan after extracting the DNA is to run a PCR, or polymerase chain reaction. Is that correct?
Exactly.
And which locus are you interested in amplifying through that process?
The cytochrome B locus. And that is a very highly conserved region of their DNA, and it's highly conserved among all kinds of rodents. And because it's very old and doesn't change as fast as other areas, we can compare how quickly they have changed. So if we're looking at one species in different areas, we can see how different they are in relation to different species in different areas.
So how did you work out in the first place that you might have contamination across individuals?
At the end of this 2-day process, after we finish extracting all of our DNA and centrifuging, and we have nice little just concentrated DNA in tiny little tubes, we run it through the biospec. And the biospec, it's off right now, but when we put a sample drop in there, it takes wavelengths from light that come in and looks for DNA, because DNA will reflect a certain wavelength. And so when we run a control, which should have no DNA in it, it should just be water and just these reagents, we have been getting readings of DNA. So that's how we can assume that there is contamination in this process.
Okay, so now you're just going through systematically and trying to work out where that is. So it could be in like your water, or even this solution here potentially.
Exactly.
So this is kind of a first step to work out. Your hypothesis is it's in the tubes.
Exactly.
All right.
So now that we have both reagents in our containers, we'll vortex slightly just to make sure everything is mixed up nice. So everything's mixed up and it should be good. We'll put it in the incubator, and this is the step where when we normally would have teeth or tissue or blood, it would dissolve overnight or over 8 hours, however long you have the opportunity to let these things melt, because we don't want solids in our chemicals when we're working with them, because we're pipetting and we need to keep things in a liquid form. So we're going to cover these with parafilm. It's a plastic sealing wrap that will prevent this from evaporating. put it in the incubator and spin it overnight so that hopefully we can have all liquid and no DNA contamination. So we'll wrap these up right now. The parafilm is hydrophobic, meaning that it doesn't like to have water in contact with it. So if we do have evaporation issues and something is leaking through the tips of these caps, this will hopefully keep that liquid inside and not let it seep out. So it's pretty malleable. Let's wrap it up nicely.
And so you're doing repeat tests?
Yes. I have been trying to find out what exactly is causing our contamination. And so I've been doing this with all kinds of different variables that I'm taking in or leaving out. So in a previous experiment, I replaced the proteinase K because I thought the proteinase K might have been contaminated. So this bottle is new. I made that last week. Or our postdoc made it. I assisted with her. We have also tried changing gloves because we thought maybe the gloves had something on them. And we will also try this experiment again to confirm that it is or it isn't the source of our contamination. Huge piece of parafoam that I cut way too big, but that's okay. Wow, that's a lot. Okay, so samples are all wrapped up. You can turn them over, heat them up, spin them around, and everything should stay in there. It'll be nice and wet on the inside and dry on the outside. So from here, we'll put it in the incubator overnight.
Nice and balanced.
Nice and balanced. We want to make sure that we place everything evenly spaced so you don't have too much weight in one side. Back. Rotate it. Incubate at 55 degrees Celsius. And that's the end of our first day of this experiment. Tomorrow we will go back and continue from this step.